Today on NBC, we will be speaking of a question that one of our viewers has asked about genetics. This question would be, if two twin brothers had children with two twin sisters, would these children be identical twins? So, throughout this newscast, we will discuss how this is highly improbable to happen.
So the three factors that make the offspring of the twins not identical is environmental, the twins' nature, and meiosis. So environmental factors makes the DNA alter. The nature of twins has modern science proving that all identical twins aren't really identical on the genetic level. And finally, meiosis, which is the gamete making process.
Various environmental factors can distort one's DNA. And if this mutation happens during meiosis, process in which sex cells are formed, it could lead to more of a diversity in the offspring. So DNA mutations work in an awkward way. First, you have to know that DNA is pretty much coded with nitrogenous bases: adenine, thymine, cytosine, and guanine. That's besides the point of knowing. The only thing that one has to get from this here is that, say, if one of the fathers or mothers was out on the beach, sunny day, let's say, and high energy radiation distorted their DNA, saying a cell that was going through meiosis to make gametes. So if you see here, when DNA replicates, it has each one of these letters codes for a different one. So your body replicates the DNA, so it would be exactly the same as its mother pair of it. So if you see here, this strand is properly replicated. But down here you can see that adenine is where guanine should be. And you can also see that cytosine is where adenine should be. So this sequence might cause different proteins to be made. Now, a protein, pretty much huge molecules inside your cells that can change the appearance that you'd have. Different people have different traits due to different proteins and their amounts. So, for example, if this mutated sequence was part of a gamete that would be fertilized or fertilize another gamete, then that offspring would have different DNA from the twins, meaning that odds are that the other twin wouldn't have had the same mutation, then the children would not have identical DNA sequences. Got that?
Done.
With there being many possibilities for the gametes of the parents to be mutated, even if they weren't, and if two unmutated gametes form together to make a zygote, there's still possibility through the vast amounts of mitosis, or cell division, pretty much to grow in size, of that zygote eventually into an embryo could happen. As you saw earlier, that was an example of a substitution DNA mutation where certain bases weren't where they're supposed to be, and it might have messed up some proteins that they coded for. This could happen in meiosis and in mitosis. So it could very well happen that an outer influence on the embryo will cause mutation, such as how much the mother exercises, what they eat, how much they eat, if they do drugs when they're pregnant, if they drink when they're pregnant, and other factors. Physical trauma, other outward factors on the mother when she's carrying the offspring will affect the DNA of the offspring and ultimately the traits. So since these two offspring are going to be having different mothers that are prone to different environmental factors, odds are is that a mutation, if it didn't even happen in the gametes, will happen in the embryo and fetus of the offspring before they're born.
So, as brought up by Caesar, the small genetic mutations that go on in everybody would make it so that the parents would not be 100% identical. So, if the parents are 100% identical to each other, then that would make it impossible for the offspring of the parents to not be 100% identical.
Meiosis. The cellular division of one diploid cell into 4 separate haploid sex cells. Diploid meaning that the cell contains 46 chromosomes, and haploid meaning that the cell only contains 23 chromosomes. Now, meiosis is split into 2 parts, meiosis 1 and meiosis 2. Now, the steps we are going to highlight to convey our point is prophase 1 and anaphase 2. Now, in prophase 1, the crossing over of chromosomes occurs. Now, the crossing over of chromosomes is when 2 chromosomes exchange like parts for one another. In this diagram, we can see how the dashed chromosomes exchange with the solid line chromosomes in the same exact parts. Now, in anaphase 2, this is when the chromosomes are pulled apart by the centromeres. When they are pulled apart, the centrioles, along with the spindle fibers, separate them into 2 separate cells. So one cell has one half of the chromatid pairs, and one cell has the other half.
So, one may ask, so what? So, if you see crossing over, to explain more in depth, doesn't happen all the time, but most likely it would happen. As you saw in the last section, you saw that a cell would have homologous chromosomes, right? One from the paternal side, and then the other from the maternal side. Alright? So, if these were to cross over, you'd get unique chromosomes with a different combination of genes than either one of the originals seen in the parents of the twins that were passed on. So just to complete that, we can see that this little section over here went right here, and it switched, right? And then here it switched too, as you can see in there. So if these chromosomes were to cross over, the gametes would be different than the cells that made up the parents. If this happens, and if the gamete goes and gets fertilized or fertilizes something, then the offspring would have a different sequence of DNA. Because as you can see, this may be the dad who has this, when the kid would only have one of these that is different. So it's different, the chromosome that's passed on. So this sparks genetic diversity, and this would lead to them not being identical. So for the fact of these chromosomes seemingly to split apart and even have a disorder among it, such as trisomy 21, trisomy X, to happen, of course the offspring would not be identical. But being more practical and probable, we can see as we have a paternal cell and a maternal cell. So you can see this
pair of chromosomes doesn't seem to be homologous. They're different, but they're grouped together as in the way how they're passed on. Now, the mother would have 2 of these X chromosomes, these sex chromosomes. So if we were to take the paternal side, right, and this would be making cells, it would have 2 possibilities of cells. It will even split that it has X and then Y in another. Now, these letters just represent the chromosomes, not their shapes. These would not be bound by centromeres to be a double chromosome, they'd be single. So, and then with the mother, you see that this would happen. Just one possibility, same proportion as that. So, in a more practical sense, one can see if these 4 parents have children. Like, this is only with one set, but if you see, you can match it up like that, and how would one know if it would be a boy or a girl that would be the offspring? So, through this way of how chromosomes split apart during meiosis, it's 50% chance, practically, that these 2 children won't have the same gender, meaning that their karyotype is not the same, meaning that their DNA is not the same, meaning that they're not identical. Now, what you see here, this sort of arrangement of possibilities that can happen from this split, even though this is the one that's most observed, because there's a direct phenotype, as you may say, of gender that comes from this type of arrangement of chromosomes being split. But this could happen with any one of the other 22 chromosomes that one could have, pairs of them, meaning that the offspring would have different chromosomes. Regardless if they cross over or not, they'll have different chromosomes, because these chromosomes, when they align in the middle of the cell, if I were to represent paternal and maternal just for each one, they don't simply align like that. The chromosomes don't know which ones are from the father of these parents and mother. They'll randomly assort or independently assort, like so. And when they split, you'll have different cells that have something that's different and unique. So if this were to bind or to fuse with another cell to make a zygote, it would be different than the original cell, because it gives 2 different types as given in this example. But this amount of different types, grows exponentially with the amount of chromosomes that a species has. So as humans, we have 23. Just imagine the different points here, all the different possibilities that can happen with just 4. But the different possibilities of sex cells that could be made in a human would actually be 2, because you have 2 chromosomes, to the 23rd power was 23 for the amount of pairs. So that is a lot of different types of assortments just through this independent assortment. This doesn't include that these chromosomes would probably be altered and crossed over as well, increasing the amount of amount of diversity in these gametes from the gametes that made the parents.
So, in conclusion, due to environmental factors, the twins not being 100% identical, and the process of meiosis, the twins' offspring would not be identical.
Thank you for tuning in this week for another pointless scientific article to make your minds melt and burn as you are in awe over the knowledge of the grandest planet in the galaxy.